Okay, let's talk about some infectious stuff. So, virioids are going to be infectious nucleic acids. The potato spindle tuber is an example of a virioid, which will infect potatoes specifically. All that is just naked nucleic acid, which will infect plants. Next, we have prions. Those are going to be a type of infectious proteins. Mad cow disease is an example of that. And what that entails is a protein that is misfolded. And when it interacts with other proteins, it may misfold those other proteins. And then when those other proteins interact with other proteins, they may infect or cause the misfolding of other proteins. And so eventually that leads to a cascade where you get cellular dysfunction and, possibly necrosis of those cells, or even apoptosis. Next, we have viruses. So viruses are going to be obligate intracellular parasites, meaning they must live within a host. Virions are what we call them when they're outside of the host. So virions is outside of the host. Once it's within the host, we call them viruses. So viruses are going to be acellular. What that implies is that there's no internal metabolism. They don't have all of the qualities of living cells. So they don't actually have a regular metabolism, so they're not going to be using ATP for any metabolic processes. In the 1890s, that's when the first virus was discovered. That was the tobacco mosaic virus, which infected tobacco. And virus or viral composition is usually going to be composed, the two bare minimum ingredients are going to be nucleic acids or their genetic material, as well as their capsid, which is going to be made out of protein, so their protein coat. So here, this green stuff, that's the genetic material, and then the outside is going to be a protein capsid. So there's going to be four different factors when describing viruses. The first is what type of genetic material do they have? Do they have DNA? Is their genetic material made out of RNA? And is that RNA or DNA single-stranded or double-stranded? So does it have single-stranded DNA or RNA or double-stranded DNA or RNA? And then next we have the protein capsid. So is it a simple capsid or is it a complex capsid? And then lastly, is the virion surrounded by a lipid membrane or not? So sometimes outside of that protein capsid, you might have a lipid membrane on the outside, on the very outside of that. Now, HIV virus is an example here. It has single-stranded RNA, and it has an icosahedral capsid, which we consider to be simple. And then it also has a membrane enshrouding its capsid, and that membrane also contains things called glycoproteins, which, if we can recall, proteins are proteins, and glyco implies some kind of sugar, and this is a mashup of sugar and protein characteristics. So bacteriophages or bacteriophages, however you want to pronounce it, those are going to be viruses that specifically infect bacteria. And within this bacteriophage example, and this could actually apply to more than just bacteriophages or bacteriophages, your viruses can undergo two different cycles. You can have a lytic cycle, which is going to be the more aggressive, damaging part of the cycle of the virus. So here's a kind of a walkthrough. So this orange color is the membrane of a bacterial cell, so the host bacterial cell. The capsid is going to be this blue tinged stuff, and then the green is going to be the genetic material of the bacteriophage or the virus. And so that virus is going to infect this bacterial cell. This purple is the genetic material of the bacterial cell. And so what happens is the phage DNA will enter the bacterial cytoplasm, and what it does is it's going to hijack the cellular machinery within the bacteria to create more of itself immediately. And so you have here, duplicates of that circular DNA from the virus, and you're creating more capsids, and you eventually just create enough of these viruses where you eventually, sometimes it could just be the physical limitations of the cell, sometimes it's an enzyme that will actually cause the breakdown of that bacterial membrane, allowing those viruses to escape and then spread to other bacteria, for example. So the phage DNA replicates and phage proteins are made, new phage particles are assembled. And so you're making more viruses and then you're sending them outside of the cell. Now, you could also undergo a lysogenic cycle if you're a virus. And a lysogenic cycle is a little bit more benign, I guess, in the sense of the integrity of the cell that it's infecting. So you have the infection of the bacterial cell. And what happens is if you're within the lysogenic cycle, what you'll do is you'll just simply integrate your DNA into the genome of the bacteria. So the phage DNA becomes incorporated into the host genome. And then every time that bacterial cell replicates, along with it, you're also replicating the genome of the virus. And so eventually it'll replicate, and you've done all the work for replicating that viral DNA. And then eventually, under stressful conditions or some kind of trigger, what will happen is it could bring the virus from the lysogenic cycle into the lytic cycle, causing the death of that cell and then releasing those viruses to the surrounding area. So the lytic cycle is going to be immediate in what we would call virulent viruses. So virulent viruses, an example of this is avian H5N1 or bird flu. So what happens is the genetic material is injected. RNA polymerase is going to attach to the viral genome promoter sequence. So that's going to be the machinery involved in transcribing DNA to RNA. So RNA polymerase from the host will attach to the viral genome promoter, and it's going to code for mRNA and therefore proteins. So proteins are going to be made that will eventually shut down the host's own transcription and stimulate only viral genome replication. And then eventually, with that machinery that you've hijacked from the host, you're going to make more of your protein coat, and then they may create an enzyme that causes the host cell to lyse, or the lysis of that host cell. And that's going to release viruses. Now, if you're undergoing the lysogenic cycle immediately, we generally consider these types of viruses to be temperate. HIV is a prime example of this, where you might not even notice the effects of the infection for 5 to 10 years later. So the viral genome is immediately inserted into hopefully for the virus, a non-disruptive position in the host genome. So we would call it a prophage at that point. So the virus's location within the bacterial genome, that would be a prophage. And then when the host replicates, the viral genome is replicated, along with that. And then under certain conditions, it may activate or trigger that virus to enter the lytic cycle, which results in, the proliferation of the virus, the breakdown of the host cell, and eventually the release of a large number of free viruses. So HIV virus is specifically going to attack your T cells, which are an important type of white blood cell in our immune system. So those T cells are going to be stored in the lymph nodes, and they'll typically multiply there. So they'll be infected, and then they'll multiply within those lymph nodes, which are all over your body. So all of these little orange packets are going to be lymph nodes. So just breaking down parts of HIV, it's got an envelope which has glycoproteins similar to T cells. And those the reason why it's able to interact so well is because of its similarities. So within its capsid, it actually has a protein that attaches to a protein that you're specifically going to find on Th cells called CD4, as well as 7 other proteins on the T cell membrane. So it specifically targets one type of cell, happens to be an immune cell. And the thing is, you know, when HIV, like if you are infected and it enters the lysogenic cycle, and you proliferate your white blood cells, over a long period of time, and eventually, maybe you reach some kind of trigger point where HIV virus is able to enter the lytic phase. Well, what will happen is, because your T cells are so vital in your immune system, you actually get a deficiency in your own immune system. So it's an acquired immune deficiency, which is pretty much considered at that point AIDS. So acquired immune deficiency. All right, so back to HIV, sorry. It's going to have single-stranded RNA as well. So here we have the single strands of RNA. Capsid is, the morphology of the capsid is perfect for the interaction between itself and T cells. All right, so a few more fun facts about HIV is it contains coding for what we would call reverse transcriptase. So A's meaning it's an enzyme. Transcript is probably going to be transcribing. This enzyme is going to be transcribing something, and it's going to be doing something in reverse. What it's going to be doing in reverse is it's going to be converting RNA into complementary DNA. That's what this little C stands for, complementary DNA. So you're actually kind of going backwards. So reverse transcriptase creates DNA out of RNA. And then another thing is, when it's creating that complementary DNA, it actually lacks good proofreading. And so there's roughly 10 errors for every 8,000 bases translated. And that's why HIV is such a hard virus to combat, because 80% of HIV patients will have different strains, because every time they multiply, you're essentially creating a new strain because it's going to be genetically different. HIV is also going to have another enzyme, or code for another enzyme, called integrase. Integrase is the enzyme, so A's meaning it's an enzyme, integra, kind of sounds like integrate. What it's going to do is it's going to integrate the HIV complementary DNA into the host DNA for its lysogenic cycle, the latent phase. So what it does is its DNA is RNA, or sorry, not its DNA, its genetic material is RNA. It recruits machinery to transcribe and translate that RNA into a protein that allows the virus to be converted from an RNA to DNA. And then the enzyme integrase integrates that DNA into the host's DNA. And then lastly, we have this enzyme called protease, or HIV-1 protease. And what that's going to do is it's going to help construct the capsid when it's eventually in that lytic cycle. So some ways we could treat HIV are through highly active anti-retroviral therapy, so HAART, and it's going to contain a protease inhibitor and some reverse transcriptase inhibitors, so it's targeting 2 different methods of your HIV replication. So it creates a protease inhibitor. Remember, protease is what's going to construct the capsid. So it inhibits the enzyme that constructs the capsid, and then it's also going to contain reverse transcriptase inhibitors. Remember, reverse transcriptase, that's going to be the enzyme that converts RNA into DNA. So those 2 things, those 2 points of interest, are usually what this 140 chemical cocktail is doing to combat the spread of HIV. Now, we could also prevent HIV The first step into knowing how to prevent it is knowing the vectors of transmission. So we've got blood, semen, vaginal secretion, open wounds, mucous membranes, childbirth, same with breast milk. So those ways we could spread HIV. And then, knowing some ways that we could prevent it through methods such as abstinence, condom use, monogamy, the use of clean needles when you're shooting up, and frequent testing of HIV, for HIV, and then simple awareness of HIV.